Hello. What we're going to do is examine chapter 26. First, I'm going to focus on the male reproductive system, and in a subsequent uh, video lecture, I'm going to do the female reproductive system. It's important to cover both of these because they play so much of a role, maybe not as much in the individual, um, in the sense that one could live a healthy life without the reproductive system to a point, but for the survival of the species, the reproductive system is critical. And as many other physiological systems exist, the reproductive systems do play a role in influencing the function of other physiological systems. Hence, they play a role in helping us achieve homeostasis. So, what we're going to do is move forward into this. Now, in the male reproductive structures, these include the external genitalia and the internal genitalia. Reproductive system, as I said, not essential for sustaining life, but the function affects other systems, and this system is essential for the survival of the species. So I have to get used to a couple of terms here and get you introduced to them to get familiarized. When I talk about gonads, these are reproductive organs. This is a generalized term. It can be either the testes or the ovaries. For gametes, we're talking about reproductive cells. So this can be the egg or the sperm. When I talk about genitalia, these are the organs of the reproductive system. For the male external genitalia, this includes the penis, scrotum, and urethra. For the male internal genitalia, this includes the ductus deferens, the seminal gland, the prostate, the bulbourethral gland, the epididymis, the spermatic cord, and of course the testes. When I talk about the reproductive tract, this is the uh, route that the sperm travel along this duct system. When I talk about semen, this is a mixture of glandular secretions and sperm cells. Okay. And if we see here from the external genitalia, the penis, urethra, scrotum, internal structures include, of course, the ductus deferens, seminal gland, prostate, which they also refer to as prostate gland, and the bulbourethral gland. Then also the epididymis and the testes. Sperm transport relies on ducts, glands, and related structures of the scrotum and testes. When sperm travel through the epididymis, the ductus deferens, the ejaculatory duct, and then the urethral to the distal part, this is the sort of transport route. We'll get more into how sperm are formed shortly. Accessory glands, these are glands that provide select fluids to protect the sperm and enhance the transfer of the sperm into the female reproductive tract. These include the seminal glands, the prostate gland, and the bulbourethral gland. When we talk about the development of the testes and positioning into the scrotum, this is a unique situation, which I'll show you in another slide here. But just to get a side view of the anatomical structures, we have, of course, the sperm formed in the testes here. They are delivered into the epididymis, which develop and get transported to the lower, almost inferior end here, and then follow along on the ductus deferens, moving all the way up around to the side of the urinary bladder and eventually come out through this area where the seminal gland is, as well as where the prostate is. Now, this ampulla is the area at the end of the ductus deferens, and basically you have this mixing here of the first secretion from the seminal gland. Then you have the semen sperm mix that continues into the prostate where there are further secretions coming here. This is the area that we call the ejaculatory duct. And then we enter into the urethra. The urethra will also at this point get a small secretion from the bulbourethral gland. And we follow the semen now, this mix of sperm and glandular secretions until it leaves at the distal part of the urethra, the urethral opening from the penis. A couple of other just minor details to keep in mind. From this point through to this point, this tract here of the urethra has both a transport capability for both semen and urine. Now, if that doesn't click, let me help you to understand that urine, of course, has a lower pH. And if there are any small droplets of urine left in the basically in this tract here, the urethral tract, they have to be somewhat neutralized so that it does not, the acidity does not affect the function or structure of the sperm cells. And we'll get into more of that later. When we go through the development, you have initially the testes which are developing, and there's a connective tissue band there. And what happens is this developing testes slides down somewhat retroperitoneal and then eventually goes down into inguinal canal here. And it develops fully into the testes in this scrotal region right here. So you can see how it kind of slides down and enters into this area. This is very important because if the testes is not in the scrotum, formation of sperm is not going to be possible, because if it's still left up in the basically the abdominal pelvic cavity, this is where this organ will not be able to successfully form sperm cells, but also this is called cryptorchism, crypto meaning hidden. The testes either have to fully descend down in there. Some cases you can surgically do this, but if they're left hidden for an excessively long time post-birth, they can possibly turn cancerous. So let's talk a little bit more about the scrotum and testes structures. In the scrotal area, you're going to have the superficial inguinal ring, which forms the entrance into the inguinal canal. The cremaster muscle, which lies deep in the dermis, it contracts during sexual arousal and in response to cool temperatures. The scrotum consists of a thin layer of skin and underlying superficial muscle. The dartos muscle is a smooth layer of muscle in the dermis of the scrotal skin. The inguinal canal really extends from the inguinal ring to the scrotal cavity. It creates a weak point in the abdominal wall. This is where you will get inguinal hernias occurring, which are protrusions of the visceral organs, particularly we're talking about like small intestine, or actually just tissue into the inguinal canal. The spermatic cords, these are the layers of fascia and muscle enclosing the ductus deferens, as well as basically composing of also blood vessels, nerves, and lymphatic vessels for the testes. And you can see them here. As you can see, this larger sheath here of the cremaster muscle, there's a thin layer of the dartos muscle, which provides some of the wrinkling that you see on the outer surface of the scrotum. And you can see the superficial inguinal ring here. You can see also the nerves and blood vessels as they come in. There is a venous plexus out here, and this is somewhat of a countercurrent exchange of heat, because really these organs, particularly the testes, have to be kept at a slightly lower temperature than the body core to allow for proper sperm development. Okay? So part of the ductus deferens, venous plexus, artery nerve, this is all makes up what we call the spermatic cord. And you have this separation here of tissue that separates the two parts of the scrotal cavity. It's called the scrotal septum. And then you have this indentation here called the raphe of the scrotum. Now, we've talked about the raphe of the scrotum. That's kind of a raised thickening of the scrotal surface. When we talk about the testes, we have several structures of importance. We have the tunica vaginalis, which is a serous membrane that lines the scrotal cavity. It reduces friction between the opposable parietal, that's the outer layer, and the visceral inner layer. You have the tunica albuginea, which is a tough fibrous capsule that covers the testes. It's continuous with the septa testes, the fibrous partitions that subdivide the testes into separate lobules. Now, these lobules are where you're going to find the seminiferous tubules. In these tubules, you have sperm production. Basically, you have 1 to 4 tubules per lobule and about 250 lobules per testes. You will also find that there are smooth muscle-like cells that contract and help transport sperm through the tubules. These muscle-like cells are called myoid cells. You also have the structure as you move up from the tubules, the rete testis, which is kind of like a little passageway or meshwork there. And sperm would leave the seminiferous tubules, go into the rete, and from the rete they would enter into the efferent ductules. And these tubules connect the rete testis to basically the epididymis, as you can see here. So you have the seminiferous tubules. They will eventually lead into the straight tubule to the rete testis. And then you'll have these other smaller tubules that eventually the sperm migrate into the epididymis where they're stored. Okay? You can see the separations of each lobule by the septa testis. Okay, and we move on. Oh, and I just wanted to clarify something there. Yep. Okay. Now, spermatogenesis. Now, this is something that should be clarified. If you've had biology before, you understand meiosis and mitosis. If you had A and P, one, you understand mitosis. In mitosis, you have the production of a cell that is basically the genetic copy of the original mother cell. So basically, you have a cell divides into two. If you started with 46 chromosomes, you end up with 46 chromosomes. Now, this process will occur, but then what happens is that you deal with gamete formation, and that has a unique property process that is called meiosis or meiosis. Okay, spermatogenesis is the term generally used for sperm production. And what we start off with is sort of like a stem cell that goes through some divisions, but eventually it starts to take on meiosis. Meiosis is only for gamete production. In spermiogenesis, this is a differentiation of immature male gametes into physically mature sperm. Sperm cells will always start off with what we call stem cells, initial cells. The cells entering the meiosis. Now, this meiosis, mind you, is a little bit different. It's what is called a reductive process. So if I had 23 pairs of chromosomes for a human cell, with a sperm cell and with an ovocell, which we'll complete next week, they have only half the chromosomes. So they only have 23 chromosomes, not 46, because 23 pairs is basically 46 chromosomes. Okay? And what you have to think about is half of your genetic material came from mom, half of it came from dad. Dad provided the sperm cell, 23 chromosomes. Mom provided the ovocell, the egg cell, 23 chromosomes. Put them together, what do you got? You got 46 chromosomes, 23 pairs. Everybody okay with that? Now, once you go through this process, it's a two-step process for meiosis. They call it a reductive process because instead of having a diploid cell, meaning you have the 23 pairs, the 46 chromosomes, you now, and that is referred to as diploid. You now go down to a new status, haploid. Okay? Haploid is only one complete set, not a pair, one complete set. So the set would be 23 chromosomes. I hope that makes it clear. So you have one diploid cell, it results in 4 haploid cells. So 4 sperm cells can come from one basic stem cell. Okay. In meiosis 1, the corresponding chromosomes from the mother and father are paired in a process called synapsis. Now, that might get you confused, but think about it this way. As I said to you before, you're a product of mom's chromosomes and dad's chromosomes. Okay, so mom provided chromosome 1, dad provided another copy of chromosome 1. Mom provided chromosome 2, dad provided another copy of chromosome 2. Okay, so we go through all of these chromosomes. And what happens in meiosis 1 is that all of the chromosome 1s, their copy and the exact original, okay? So they're all paired together, all the chromosome 1s, both moms and dads. And what happens is that you form this what is called synapsis. This pairing up is the process called synapsis. But what you see is a matched set of 4 chromosomes. Because before you go through meiosis, you have mitosis. Okay? You doubled up your copy. The first step in meiosis 1 is you're going from that paired up double copy down to one set of copies, and then you go to haploid. And you'll see this on the spermatogenesis. The bottom line is, after you've gone through all of this replication, you end up with each reproductive cell, each gamete, whether it's sperm or egg, being able to have only a haploid number. Now, to help you understand this, they call it the daughter cell. So at this point, we're at 2n. But of course, what starts off everything is you're going to have to take all of the chromosome 1s and all, both from mom and dad, as well as the copies that you've made. And then you're going to go through this reductive process, the synapsis, the tetrad formation, then you start spreading them out, and eventually you get down to only half of the number of chromosomes that are in your normal body cells. 23 chromosomes. That's haploid. Now, if you see what happens here, you start off, you reproduce everything, then you start reducing this all. So this allows you to form sperm cells. Now, the sperm cells, by the way, as you go through these levels of meiosis, the second level in particular, you're going to start having some possibility of mixing. So there might be a sperm cell that's got mom's number 1, dad's number 2, mom's number 3, dad's number 4, mom's number 5, and number 6 chromosome. So some sperm, you ever had when somebody looks at a particular kid and they say, you look just like your mom, you look just like your dad, etc. Well, some of these external attributes are because you had some more of dad or mom, etc. But the bottom line is that the sperm cell is a reproductive cell. It has to still meet with the egg cell to form an embryo, a zygote, a fertilized egg. Okay? That we'll get into in a subsequent lesson. But let's focus back into the seminiferous tubules for sperm formation, spermogenesis. Okay? When we talk about spermogenesis, this is the process that takes 24 days. This is from spermatids to mature sperm. Let's not forget about what's the structure of sperm cells. On the tip of them, they have this membranous compartment. It contains enzymes essential for fertilization. It's called the acrosome. The acrosome is absolutely necessary so that it can allow for attachment and then sort of this enzymatic dissolving getting through the outer layer most of the egg cell to create the fusion for the sperm cell to meet in with the egg cell. Again, we'll get more into that later. We have the head, which contains the nucleus with densely packed chromosomes. We have the neck that contains both centrioles of the original spermatid. And then we have microtubules of the distal centriole are continuous with those of the middle piece and the tail. So there's some construction structures here. The big one that's really important that I want you to keep in mind is the middle piece. Why? Contains lots of mitochondria. Why are there mitochondria there? To make ATP. Why is ATP necessary? Because you're going to need the energy to run the flagellum. That's the tail. That's going to be whipping back and forth. That's your propulsion for the sperm cell. So you have to keep in mind, no mitochondria, no ATP production, no transport, no propulsion of that sperm cell. Here is an example of that acrosome, head, neck, middle piece, and tail. Here is a picture of healthy sperm on the right. Here is a picture of not so healthy cells. Everybody talks about this, but I think this is a decent diagram because it really helps you to understand. Any cells that are abnormal are going to either not be successful in transporting through the female reproductive system, or unsuccessful in fertilization, or lead to a defective embryo if they are able to successfully fertilize at all. These are normal. This is a giant one, so obviously this doesn't look like it would make it. This is a microsperm, so basically you barely have anything in the head region. You have a double-headed one here. I know, two heads are better than one. No, not in this case. This would just lead to what might be considered trisomy, or some type of 3 times the number of chromosomes. That would be a mess. Double body, and you can see here they would have double of the neck and middle piece. This would probably also fail. Long head, rough head, abnormal middle piece. You can see the wiggle in it. But all of these would be unsuccessful in one sense or another in fertilization. Now, meiosis and early spermatogenesis occur within the seminiferous tubules, which I've talked about. The rete testi is a network of canals. The septa testi separates the lobules. The sperm development in the seminiferous tubules is from the walls of the seminiferous tubule toward the lumen. Think about the lumen as the center. The center is clear in a lot of images. You'll also see a bunch of little tiny sperms cells, mature sperm cells in the lumen. It's the center. So you're moving from wall toward the center of the tubule. And as you move through this, you're going through several levels of development and specialization. Now, the tubules are surrounded by a delicate connective tissue capsule and areolar tissue, filling the spaces between the tubules. You have also present interstitial cells, otherwise known as Leydig cells. These produce androgens, such as testosterone and androstenedione. These are the sex hormones. You have Sertoli cells, which are also called nurse cells. They're attached to the tubular capsule. They extend to the lumen. The cytoplasm will surround, the cytoplasm of the nerve cells, that is. They're going to surround the developing spermatocytes and they're going to assist it. That's why they are called nurse cells. They help in the nurturing of this. You can see from this tissue slide here, you have the seminiferous tubules here. If you up the magnification, you can see here in the center is the lumen. And this is a complete seminiferous tubule. You will see that dark shading here. That is going to be sperm that are in the lumen here. So everything is going to be developing from the wall toward the center. Again, from the wall toward the center, and you can see cells in various stages of division, of going through meiosis, and of getting nurtured by the nurse cells. Excuse me. Now, interestingly enough, you can see the nurse cell nucleus here. They will have tight junctions around themselves. And what happens is basically you can see that the sperm cells go through various levels of development, eventually lose most of their cytoplasm, and have a tightening up of the DNA in the nucleus so that's fitting in the head, and eventually get the nutrients necessary to form the flagella. And then they would be hanging out here in the lumen when they're completely mature at this stage. Okay? Now, moving on, we have the terms spermiation, which is the sperm cell loses attachment to the nurse cell and enters the lumen of the seminiferous tubule. Now, there's one other very interesting situation here that we want to bring up. That's the blood-testis barrier. This basically isolates the antigens of the seminiferous tubules and the sperm cells from the blood. You see, the nurse cells with tight junctions basically are separating the blood from the cells. So this is another one of these tissue blood barriers that you see. We've talked about the blood-brain barrier. We've talked about the blood-thymus barrier. We've got one here, the blood-testis barrier. This is very important. You got to keep in mind also nurse cells have basal compartment, which is an outer layer, and an inner luminal compartment where meiosis and spermatogenesis occurs. Okay? Now, look over around here. You've got all these tight junctions, etc. So basically, there is no association of blood with the antigens and proteins here. You might say to yourself, so what? Well, let me just give you a couple of the pointers here. Look, as you're going through this development here, you're noticing from primary to secondary spermatocyte, eventually forming spermatotids, beginning the spermiogenesis, etc. What's going on? You're turning off on certain genes, you're turning on certain genes, you're making proteins, aka antigens, that have not been seen by the immune system at all. So what you have is the potential that if the blood system is exposed to these antigens out here, you're going to have an immune reaction. Keep in mind, these nurse cells provide the nutrients necessary, the signals necessary for proper cell development here. Okay? Before I go there, what I want to do is just bring up this point. When males go through a vasectomy, you basically are cutting somewhere in there the ductus deferens. It's also called the vas deferens. And there have been individuals who have this procedure done, and they clip a small amount. They may cauterize one of the ends, etc. But what happens inevitably is that blood cells start going down, particularly white blood cells, down the vas deferens, down the ductus deferens is the other name for it, eventually into the epididymis, and then eventually into the seminiferous tubules. And that's where you get an immune reaction. You will eventually form antibodies, and you may have, as some males have reported, really painful inflammation. And it's not like they can just go out with their bowling buddies or their golf buddies after they've had a vasectomy. They may have an adversive immune reaction that occurs. Also, the statistics of reversals of vasectomies, which some individuals have tried to do, the established papers have stated clearly that usually the rate of success of a reversal, sorry about that, declines greatly as time goes on. What I'm basically saying is that the success of a reversal after 1 year is less than 50%, but by the time you're at 5 years, it's just virtually almost zero. And the reason being is that even if you do produce sperm after this reversal, they may be coated with antibodies, which would inhibit their capacity to fertilize an egg cell. Okay? Just I bring those things up. Now, when we talk about the male reproductive tract receiving secretions from the various glands, seminal, prostate, bulbal, urethral, we have to look at what's going on and why. We have the term capacitation, which is the process that causes sperm to become mobile. And what I mean by that is they become actively swimming and they become also at the same time fully functional. This is a two-step process. Sperm become motile, when mixed with seminal gland secretions. The sperm become capable of successful fertilization when exposed to conditions of the female reproductive tract. So there's certain chemical signals that are sent to cause motility, and then there are other chemical signals present in the female reproductive tract that enhances fertilization. As we've been moving through the ductus deferens, there is sort of an expanded portion called the ampulla. After the ampulla, one goes into an area called the ejaculatory duct, which carries fluid from the seminal gland and ampulla to the urethra. As you can see here, we have the ductus deferens, so the sperm here would be produced down here and traveling down through this area. And then we get to the ampulla here, and then we get into this area called the ejaculatory duct. Here they're getting secretions from the seminal gland. The seminal gland will also produce a variety of compounds. I believe one of them is fructose, which acts as a fuel, which the mitochondria would use to provide ATP to cause motility, the movement of the flagella. Okay? We will get other secretions coming in from the prostate gland here as we empty into the prostatic urethra. Finally, as we continue down through the urethra, we get final secretions from these small glands called the bulbourethral glands. So how do we start this journey? We started here in the epididymis. If you notice, the epididymis is long, coiled, etc. Starts off at the superior part of the testes, comes down to about the inferior part, and then moves into the ductus deferens. This is mostly a storage site for sperm cells. You can see the sperm in the lumen. You can see the walls of the epididymis, and the epithelium is here. There's also some extremely long structures called the sterocilia. They're not really cilia per se, but they are important in passing on nutrients as the sperm cells are basically, shall we say, waiting there. Now, the epididymis is the start of the male reproductive tract. It's a coiled tube to store sperm cells, as I've said. It consists of the head, which receives the sperm from the efferent ductilus, body, which extends inferior along the posterior surface of the testes, and a tail, which is the inferior border of the testes. This connects with the ductus deferens. The epididymis is lined with pseudostratified columnar epithelial lining with long sterocilia, which acts to enhance absorption from and secretion into the fluid of the tubule. Now, the ductus deferens can store sperm in the ampulla for several months. This transports the sperm from the epididymis to the ejaculatory duct. The seminal glands, known as seminal vesicles also, produce 60% of the volume of semen. Once you get to the point where you start adding on fluids, you're changing your terminology. Yes, you have sperm cells, but it's mixed with these secretions, and so they refer to it as semen. The secretions at the seminal glands will activate the sperm flagella. The prostate, which is a small muscular rounded organ, will also produce secretions. These include seminal plasmin, which is an antibiotic protein that helps prevent infections in males. Okay. Then we have the bulbourethral gland, which also has the name Cowper glands, also known as Cowper glands. And they're located at the base of the penis, and they secrete a thick alkaline mucus. This helps to neutralize any urinary acidity that may remain in the urethra. It also will lubricate the tip of the penis. So here we have the ductus deferens. You can see the layer of thick, smooth muscle here. That would, of course, allow for peristalsis, the transport of the sperm down through here. Then we have the seminal gland, which has a lot of secretory pockets and also smooth muscle on the outer layers. The prostate also has connective tissue and smooth muscle, but it also has a lot of tubulo-alveolar glands. If you're not familiar with that, go back to chapter 4 in the histology, basically the way the tube structures, the glands are arranged. And finally, we have the very small but important bulbourethral gland, which you can see the lumen there, as well as mucus glands and the smooth muscle, and of course the capsule. The point being is that the bulbourethra will basically provide a protective coating, a neutralizing coating, as the semen is transported through the urethra. Okay. Now the penis conducts urine and semen to the exterior. The penis is defined as a tubular organ for sexual intercourse and for conducting urine to the exterior. The structure of the penis is the root, which is the fixed portion that attaches the penis to the body wall. The body of the penis, which is the tubular movable portion of the organ. And then we have the glans penis, which is also referred to as the head. It's the expanded distal end that surrounds the external urethral orifice. The neck of the penis is a narrow portion of the penis between the shaft and the glans. And then we have, of course, the erectile tissue, which consists of 3 cylindrical columns of vascularized tissue. Okay. Now, in the resting state, these highly vascularized cylindrical structures, the arterial branches are constricted and the muscular partitions are tense. During an erection, the penis will stiffen and elevate to an upright position, and the arterial branches were going to be dilated. The venous sinuses will narrow. Now, this is where they retain the blood to maintain the erection. So when you look at the cavernous or the corpora, these vascularized regions, you have 2 of them that are called the corpora cavernosa. These extend along the length of the penis as far as up to the neck. But then you have the corpora spongiosum, which surrounds the penis. It's an erectile tissue that extends from the tip of the penis to expand and form the glands of the penis. And what I mean by it surrounds the penis, really what it is it's surrounding a urethra. Excuse me there. So the corpora spongiosum really surrounds the urethra. If I go back here for a second, I'll go advanced one more second here. You have the corpus spongiosum down here, and you have the 2 cavernosa here to here. One more thing. We have a variety of erectile dysfunction drugs that are out there. Cialis and the other ones, Viagra, etc. Now, one of the things, if you listen carefully to the commercials, and they will insert along a little quiet voice, if you have erections for longer than 4 hours, you should seek out your doctor. What happens is that these are sort of like the fine print in a contract, but they're the warning parts. Here's part of what you have to think about. You have arterial blood going into an area and being almost held or sequestered for a period of time in these large venous sinus areas. Okay. Now, there is a disorder called preism, where an individual has an extended period of time of having an erection. Now, the part about the 4 hours is that if by taking these drugs, the individual has this for an extended period of time, surgeons have to go in and remove the blood because it will start to cause literally tissue to become gangrenous. And although one could make lots of jokes and humor and everything else, the reality is this is a medical condition. Sometimes it will happen when individuals use these erectile dysfunction drugs but don't deal with the situation in an intelligent manner. And so what happens is the blood tends to pool there and stay in this erectile tissue, but eventually starts becoming a problem because there's no oxygenation, and so the tissue will begin to die. Okay, so let me move from there to the tissue layers of the penis. Of course, the skin, which is the overlying skin of the penis, is very similar to the scrotum. The dermis, which has a layer of smooth muscle continuous with the dartos muscle. Underneath that, you have the areolar tissue, which allows the thin skin to move without distorting deeper structures. You have also a dense network of elastic fibers. These encircle the internal structures of the penis. Now, there's a structure that in some cultures is seen and others is not. It's called the foreskin, otherwise known as the prepuce. This is a fold of skin that surrounds the tip of the penis, attaches to the narrow neck of the penis, and continues over the glans penis. Preputial glands in the skin of the neck and on the inner surface of the prepuce produce this waxy material known as smegma. This can lead to certain infections and therefore, for medical health reasons, as well as cultural, religious, philosophical, a lot of times the foreskins are removed after birth for males. Okay, so the foreskin would be around in this area, right through here over the glans penis. Now, here you see the example of the erectile tissues. You have the corpus cavernosa here, and you have the corpus spongiosum, which really encloses and surrounds the urethra. Here's a deep artery of the penis here and here. Excuse me. You have some areolar tissue here, and you'll notice also that you have some dorsal veins, arteries, etc., and nerves. Human sexual function during the sexual response consists of the following: arousal, emission, ejaculation. In the arousal state, you have stimuli, either thoughts or sensory sensations carried by sensory nerves from the genital region, and they result in a parasympathetic outflow over the pelvic nerves. Now, nitric oxide neurons play a role in the vasodilation of the penile arteries. This is where the drugs, Cialis and Viagra and all these play a role. They help maintain the nitric oxide neurons' function by enhancing and elongating the signal. These drugs block phosphodiesterase 5 from breaking down the signal, so it maintains itself much longer. We then have emission, which upon further stimulation leads to the sympathetic activation that leads to the emission. That's the transport of semen through the ductus deferens and the urethra. And then we have ejaculation, which occurs as rhythmic contractions occur in the ischiocavernosus and bulbospongiosus muscles. These are controlled by the somatic motor neurons. Associated with them will be, with the contractions, will be pleasurable sensations known as the male orgasm. And you can see how this would work through here. Okay, now let's get into the endocrinology of this. Testosterone plays a key role in establishing and maintaining the male sexual function. Testosterone is produced by the interstitial endocrine cells of the testes. But testosterone is involved in a negative feedback loop to maintain and to control the amount produced. First off, we look at the hypothalamus, which releases gonadotropin-releasing hormone, GnRH. This hormone acts on the pituitary cells. What it's going to do is stimulate from these anterior pituitary cells. Some of the cells will release luteinizing hormone, LH, as well as follicle stimulating hormone, FSH. Now, FSH targets nurse cells in the seminiferous tubules. LH targets interstitial endocrine cells of the testes. So what you're talking about there is FSH is really working on the development indirectly of the cells that are going to play a key role in spermatogenesis. LH is going to provide stimulation to the cells that actually produce testosterone. Now, LH is going to induce a secretion of both testosterone and other androgens. It's testosterone that negatively feeds back to inhibit the GnRH. So there's the negative feedback there. So you have testosterone feeding back to the hypothalamus, acting to control the release of GnRH, which then would have a control on FSH and LH secretion, as you can see here on this diagram here. But wait, there's a little bit more. Because we have nurse cells that promote the spermatogenesis and spermiogenesis, and they're going to secrete androgen binding protein, otherwise known as androgen binding hormone protein. ABP binds androgens. Well, that only makes sense, and mostly testosterone, in the fluid of the seminiferous tubules. Now, keep in mind something here. Testosterone, which its basic structure is very similar to cholesterol. So that puts it in the lipid category, which means that lipid is not very soluble in a watery environment. It needs assistance, in this case, by a particular protein that will bind to it and help it to remain in a certain concentration in the fluid area of the seminiferous tubules. Now, of course, this is going to increase the concentration of androgens and stimulate physical maturation of the spermatids. Nurse cells will also release a protein hormone by the name of inhibin. This is released in response to factors released by developing sperm. What does inhibin do? Well, it happens to, interestingly enough, depress pituitary production of FSH. Now, there's some debate whether it may influence GnRH or not. And the jury's not totally in on that to be completely convinced. But here is one interesting thing. Inhibin may provide merely a feedback control of sperm production. So basically you have testosterone, which has the overwhelming negative feedback control right back to the hypothalamus, and thereby in this cascade reduce GnRH and FSH and LH. But inhibin will depress the pituitary production of FSH, which will then control sperm production. And you can see this all mentioned out here. Now, before we leave, we wanted to look at reproductive disorders. Again, I've taken chapter 26 and just split it from male and then female. So this was part of the latter part of the chapter 26, but I focus it only on the male reproductive system. When you look at benign prostatic hypertrophy, what we're talking about is affecting the prostate. It is typically a disorder that will occur in men after age 40, usually occurs as testosterone production by the interstitial endocrine cells will decline. Benign meaning it's not malignant, but you start having the prostate beginning to get bigger and bigger. And as it does, it may, in some cases, block urinary flow, because as it gets bigger, it's putting more pressure on the urethra, the prostatic urethra, and may inhibit or block restricts some of the urinary flow. This differs from prostate cancer. This is a malignancy of the prostate. It is the most common form of cancer death in males. It can be monitored and detected with a blood test that looks for prostate-specific antigen, PSA. And usually, men will get a test as part of the basic blood panel, blood tests, and it's fairly sensitive to indicate for an antigen that is, if it is elevated, it's going to be an indicator of prostate cancer. If it way swings up, that's the next sign. The doctor then has to go in and look for possibly prostate cancer. Usually, if you're monitoring year to year, et cetera, you'll be able to keep a good handle on things and then move forward if something develops later. Then we have finally testicular cancer, which is the cancer of the testicles. The average age of diagnosis of testicular cancer is 33. 95% of this is due to abnormal spermatogonia or spermatocytes. In other words, it's not nerve cells, it's not the seminiferous tubule wall cells or anything else like that. It's usually some abnormal spermatogonia that somehow went haywire. And usually the treatment consists of an orectomy, the removal of the testes, and maybe chemotherapy. You can see how as this prostate is enlarging, it's narrowing and constricting the prostatic urethra, and therefore it would make it very hard for urine flow for the bladder to completely empty itself. And this is the situation here where you have cancerous tissue developing in the testes, and basically they would have to remove the testes surgically. And so we peruse the chapter review outline on pages 1031 to 1033. Complete the review questions of 1034 to 1035 and complete the review sheet on 1012. One suggestion is this: after you're done with this lecture, once I put up the female reproductive, then the review outline and the review questions at the end of the chapter will probably be able to handle both sets. What we're going to do for the next lecture is continue chapter 26 on the reproductive system for the female.